I would like our first speaker, our first panelist, Joanna Bonds, to introduce herself, just give us like 2-minute self-introduction. And yeah, thank you.
Thank you, Alexandra. Good morning, good afternoon, everyone. I am Joanna Bonds. I'm currently a postdoc in the laboratory of Dr. Birgit Schilling at the Buck Institute in the Bay Area. And before I joined the lab of Dr. Schilling, like 1.5 years ago, I was doing my PhD in France in the laboratory of bioorganic mass spectrometry under the supervision of Dr. Christine Carapito. And my PhD was focused on the development of quantitative proteomics methods applied to various collaborative projects. And one project I was involved in was about food proteomics. And this project was done in collaboration with Dr. Muriel Bonnet and Brigitte Picard, who are working at the French Institute of Agronomical Research. And the aim of this project was or is to develop a tool that would help to predict beef meat quality traits, namely tenderness and adiposity of beef meat. And for this purpose, they wanted to use protein biomarkers at the basement of the tool to predict it. And so in this regard, I was involved in developing PRM targeted methods in order to verify biomarker candidate of beef meat tenderness and adiposity. So this is how I was involved in a food proteomics project.
Sounds very exciting. I can't wait to hear a little bit more about this later on in our question session. Well, I will go alphabetically, so I will ask now Corey to introduce himself.
Hello, everyone. My name is Corey. It's really nice to be here today. Alexandra was kind enough to kind of keep me in mind and extend the invitation to join the group today. So I'll have to say, I've been involved in mass spectrometry-based protein analysis for a long time. But in terms of my connection to food science, even though I've been in the human health area for a very, kind of most of my career, I started out my life as a food scientist. So I have a bachelor's and a master's degree in food chemistry from the University of Wisconsin in Madison. And after my postdoctoral work, I actually spent 4 years working in New Zealand at Fonterra. And that is now recognized as probably the world's largest dairy company. And so I always kind of have to pause with these pieces, right? Which is, okay, so you're making milk and cheese and yogurt. But the reality is that, most food companies are integrated technologically. And so for a company like Fonterra, there was a microbial physiology group very interested in what we now call microbiome work. So the early thinking about beneficial microorganisms, the physiology of microorganisms that are used to produce cheese and cheese cultures and yogurt cultures. There was a very robust animal physiology group. So looking at the fundamental metabolism that's required for animals to mobilize enormous amounts of protein and calcium into the mammary gland to basically make the raw material that's produced into the cheese, yogurt, and foods that we love. And we also did a lot of kind of product, kind of very product focused types of work. Took a long break in my career and then recently had a reintroduction to the food tech world. I had a brief stint at Impossible Foods. And I think most folks who kind of watch the space a little bit are aware that there's a lot of interest in plant-based meat substitutes, and Impossible Foods is one of the companies that competes in that area. So I'll be happy, through the course of our conversation today to talk a little bit about how mass spectrometry and other analytical chemistry technologies are being used at the highest levels in the food industry and the kind of interesting opportunities that are out there for people exploring careers in that space. So thanks, Alexandra. It's really nice to be invited to be here today.
Well, thank you so much for accepting the invitation. I think you have a very broad view on all subtypes of avenues that you can take after bioscience career. So I think there will be some good questions for you to answer and give a broader overview as well to the audience today. Now I would like Melanie to introduce herself.
Sure. Thanks, Alexandra. I'm Melanie Downs, and I am an assistant professor in the Department of Food Science and Technology at the University of Nebraska-Lincoln. I'm also affiliated with a group here in the department called the Food Allergy Research and Resource Program. And so as you may imagine from that title, a lot of my work, my research work focuses on food allergens. And we're particularly interested in, the ways that food allergen control and management impacts the food industry. So we, on the research side, we do a fair amount of work looking at development of quantitative methods for food allergens. And so in the past, that was mostly immuno-based methodologies, but now most of the research is focused on proteomics methods, targeted quantitative proteomics methods to detect food allergens in different types of processed food matrices. Because almost all food allergens of interest are proteins, in case that wasn't obvious, but so that's why we're interested in the proteomic applications. We're also interested in, ways that we can use proteomics to characterize food allergens. There's some foods where less is known about the proteins that are allergenic than you might think. We're also interested in ways that food processing can change food allergens. Change their structure, change the profile and abundance of different proteins in different products. Similar to what Corey was discussing before, there's also lots of food processing operations, fermentation, et cetera, that maybe partially hydrolyze proteins or change them in some way. So we're interested in characterizing the proteins that could be food allergens after that. We also have some additional work looking at novel protein sources and trying to evaluate whether those are likely to be allergenic sources, as well as, how similar are these novel proteins to known and relevant food allergens. So a number of different areas, mostly anything related to food proteins is kind of within our allergen wheelhouse as well. So happy to be here and glad to answer any other questions.
We are so happy to have you here. I'm already have at least 3 questions popping into my head just while you were introducing yourself. So yeah, I can't wait to start our discussion. Okay, and our final panelist, Jamie, please introduce yourself.
Hello. Let's see, how does this? I'm Jamie Kaler, Senior Research Chemist at Cargill. I've worn many hats at Cargill. I actually brought in advanced proteomics into Cargill. I've been working on it for the past 3 years, probably. As you can imagine, it's kind of difficult to bring in new technology to a company as large as Cargill when our research footprint really isn't as big, but it's bigger than you think it is, but it's not quite as large as some companies that focus completely on research. I've touched a lot of things at Cargill, all the way from basically fermentation monitoring, organic acids, sugars, alcohols. Cargill is a large food company where we have lots of industrial fermentation, citric, adipic, ethanol, name it. I got away from analytics for a while, actually did some engineering work at a really exciting project several years ago, got back into analytics, and really work to do sort of advanced method development, advanced method troubleshooting with our plants, with and for our plants. The last 5 years has really been about bringing in new technology. So coming up with what our strategy should be, selling that capability to management and really bringing it in-house. Proteomics has been a ton of fun since we brought in the instrument we brought in the capability about 2 years ago. Our primary focus areas really are supporting the biotech team, right? So molecular biology, fermentation monitoring, where's the carbon going? Are they engineering the right pathway enzymes? Are they there in the amount that they think they are between strains? identification of proteins. I think I heard someone say, I don't remember who, I think many folks on the plant panel had mentioned alternative proteins. As you can imagine, Cargill is very interested in alternative proteins. So I think that's where a lot of our work, my team's work, will go in the next several years. I guess the other piece would really be in that animal health space. We've acquired, I'm sure you followed some of our acquisitions, and if you haven't, we've been interested in really getting into sort of how some of our fermentation waste streams sort of really help out the health of animals, companion animals, ruminants, pigs, any farm animal that you can really think of, and maybe potentially branching off into humans. So we're looking at that microbiome, understanding how different feed additives and different feeds that either we sell or other people sell really affect that overall microbiome and therefore leading to animal health. I love science. I think that this platform is amazing. I'm very grateful to have been asked, to join. I wish it was available when I was early career. So happy to answer any questions anyone has. I'm really looking forward to the discussion today.
Awesome. Thank you so much. It seems that you can do a lot of different things there at Cargill. Okay, so I'm really happy to open the discussion officially. So every participant is highly encouraged to drop any questions they have to the speakers. However, I would like to start with two questions that I would like all of the panelists to address. Please feel free to keep it a casual discussion, casual conversation, interact with each other, and please start by telling us what type of work do you do every day on your typical day, and I would like you to tell us and our participants 2 top things that you wish you knew before you started working in the food sector. So let's start maybe with Melanie.
Okay, well, I think I'm the sole academic on the call. And so, having been a PhD student, you may have known what your PI did. Maybe you didn't know what they did all day, but I'll say for me, so, there are different types of faculty appointments. University of Nebraska is a land-grant university, so I have kind of a unique appointment in that I have some research responsibilities, some teaching responsibilities, and then also some extension responsibilities. So if you're not familiar with extension, it's a program at all land-grant universities. It's really intended to communicate and transmit science-based practices to end users, right? And the original model was for, agricultural science to farmers in counties, right? But it's much broader than that these days. There's lots of different types of extension activities. Ours in particular is a lot of our work with the food industry on various food safety concerns, food allergens for us in particular. So my day-to-day activities generally involve, most of the time there's some sort of meeting with grad students that I supervise to talk about data, how their projects are going, that sort of thing. So I supervise anywhere from 4 to 6 graduate students, oftentimes co-supervised with someone else. And so, generally we have some activities to look at how their research is going. Also generally have some research planning that you're thinking about, right? Whether it's grant writing, publication writing, those sorts of activities. My teaching depends a little bit upon the semester, right? I have a smaller teaching appointment, but obviously that includes preparation for classes, these days recording lectures, but also presenting live lectures, right? So that's a pretty common activity. And then on the extension side, that's where things are, a little bit less predictable in some cases, right? So in some cases, you're fielding questions that come in from the food industry, right? They may have a food allergen management question, a detection method question, those sorts of things. So doing that, but then also planning programming. So planning, training, and education activities. So we actively go out to those stakeholders as well and do training activities. So I think that probably encapsulates a lot of what I do. Obviously, there's also the administrative stuff that everybody loves that also comes along with a faculty position. I think one of the things I wish I would have known and maybe would have, maybe tried to work a little bit harder to get some good habits going is how much writing is required as a faculty member.
Oh yes,
it's
that's a big part of that academic life, right?
And I think, as you become a faculty, you spend almost no time in the lab, to be honest, and most of your time is writing grant proposals, writing and reviewing publications, things like that. So, if you're interested in becoming a faculty member, definitely think about, whether you like writing. If you hate writing, you might struggle being a faculty member, but also, trying to get some experience and some, helpful things that work for you and to be able to do a fair amount of writing activity. So.
Thanks so much.
Yeah.
All right. Maybe now let's move to Joanna. Joanna.
Thank you. So I would say for me it was a bit different because I was involved in food proteomics as a project during my PhD. So really my role was to get introduced with the collaborators into the projects, understand what was the aim of the project and what they wanted us to do. So namely, our part in this project was to absolutely quantify 10 protein biomarkers or biomarker candidates for beef meat quality traits. So my role there was to develop the assay, to prepare the samples, run the samples, then analyze the data and report to the collaborators the results. And so they were then able to interpret the data and see, okay, what if it makes sense, because we analyzed the samples in a large cohort of bovine muscle tissues that was spanning a very wide dynamic range of tenderness and adiposity in order to verify the potential of the biomarkers. So they were then able to assess whether it was suitable to keep such proteins for further investigations in order to develop their tool. So I was really at the bench and the mass spec and data analysis part, and then interacting with the collaborators about the results, and then writing a manuscript about the results and to further continue.
Okay, thank you so much. Let's talk about Jamie's experiences now.
Hi, everybody again. Yes, so at Cargill, what does my day look like? So as the proteomic subject matter expert, as the proteomic capability lead at Cargill, I'm in charge of sort of method development, method validation, training more junior scientists. Proteomic sample prep, as you know, is really complicated. It's multifaceted, pretty iterative. So it requires, I think, a really in-depth understanding of what you're doing with each, I call them sample prep unit ops. So we spend a lot of time training scientists in how to, why are we doing what we're doing? Why do you have to apply these, salting out the protein? Why do you have to clean it up? Why do you have to digest it? So that's kind of a lot of time. I love training. I love teaching scientists new things. So I think to what Melanie was saying, maybe I should be in academia. I don't know, but I do leverage that capability or that a lot at Cargill. I'm also in charge of making sure that all the reports that go out with our collaborators are accurate and reflective of sort of what the collaborator was expecting versus what the data shows a lot of times. Report writing, but also what I think my role here at Cargill has really branched into really is selling the capability. When you're in a company as large as Cargill, where you have a fairly small research but impactful research group, getting the word out to the power of proteomics. And I tell people that, 5 years ago, I couldn't have imagined a company like Cargill diving into proteomics, $1.5 million mass specs, all the work that it entails. But I think I'm pretty good at marketing, pretty good at sales, but also being really technically deep. So that's another skill that I think I've leveraged at Cargill to really make sure everyone, even across their technical acumen, really understands the power of proteomics and what it can and what it can't do for you. So I'm also charged with doing presentations a lot, all the way up to upper management at this behemoth of a company. But every day we learn something new, and every day we're driving forward the capability. One of the other things, like I said, to get capability expansion, whereas we focus a lot of time on bottom-up proteomics, the next focus will be sort of top-down intact protein analysis, looking for post-translational modifications and such. What do I wish I had known before diving headlong into science and or proteomics was really sort of how difficult it can be sometimes to sort of sell technology to a company that's not technology-based. And having patience and really leveraging your sort of political skills and political acumen. And then, of course, and how quick people expect you to turn around data, right? So you bring in capability within 3 months, you better darn well have several examples of how you led to some more insight for the company. So that's what I wish I had known.
Thank you so much. I almost want to say that I wish in all fields we had the previous knowledge about that. Is so useful. Okay, and finally, Corey, what's your everyday responsibilities, and what's what you wish you knew before you went into food industry?
Sure. So everyday responsibilities is a great question, because I think, these are, at least from my perspective, because I'm not in the food science field right now, generic, and I pivot a lot on what Jamie has kind of indicated in terms of commercial environment activities. I view my job, in a more senior role, is connecting science to fundamental paths to answer commercially relevant questions and try to do the best job to keep that connection. It's really easy. And I think, as scientists, we always love the details. We like to really get into the finest points. But at the end of the day, we're looking for the broad brush, impactful data that's going to allow our companies to move forward, sell improved products, sell more products, or break into new areas. And so I think that's been a common thread, whether I've been in pharma, I've been in diagnostics, or I've been in food science, right? So I think you can see those things everywhere. And maybe a point that Jamie takes or has made already is that, particularly in the food area, which is largely often kind of considered a commodity-driven business, you're probably really a lot closer to having to make hard commercial decisions. You have to really, really push. The deep details, for what is relevant moves the dial for a company like a Cargill or an M&M Mars or a General Foods is much, much different than what moves the dial for a Pfizer or a Merck or kind of a biopharmaceutical company. So I think that's kind of really a common thread. A couple of things that I guess I wish that I knew, and I guess part of the thing is the world has changed so much, and so quickly. But if I go all the way back to when I was in undergraduate, studying food science for my bachelor's and my master's degree, I think I didn't even at the time understand the level that science was being performed at the very best companies. You get little hints of that, if you get a chance and you're in a food science department. But I think a lot of people who, kind of maybe get a PhD at a top-notch academic institution in proteomics or kind of molecular biology, these things, don't even recognize that a company like Nestle, right, has got fundamental interest in doing science at the same level that you'd see at the top-notch academic institution. So that was only something. That was only something that I've learned. And I think the other thing I would just point to directionally is that it's a brave new world right now, right? Because with the advent of food technology companies, right, they're kind of inverting this whole thing and saying, well, food isn't it's not a commodity. Our job is to, kind of bring the highest technology, science, and marketing to kind of work the other direction. So, these food tech companies are starting up with the idea of having proteomics and metabolomics and doing all of these things in a way that's very, very different. So I guess, if maybe there was something I wish I knew from when I was really young, is how wildly the world can change, over the course of a career.
Oh, that's so true. All right, thank you so much. I have first question from the Q&A box. Nathan Bassisti is asking our panelists, what are the job career prospects for trainees interested in getting into food science or food proteomics? Are most opportunities either in the industry or rather government grant funding for this type of research? Anyone who would like to answer this question first? Okay, Jamie, please.
Yeah, I think that there's, like I said during a few minutes ago, there's tons of opportunities in Cargill, for example, and I'm sure in industry-wide, we're growing in this space. The more we're just, tipping our feet into the sort of proteomic space, but the more and more, it's like a snowball, right? The more and more we show that there's value. In fact, I think in the next couple of years, I think across the omics space in general, we've been asked for like 4 FTEs, and guess what? There's only 2 FTEs right now working on it. So I would say there's tons of opportunities, and it's only going to grow for us at Cargill, and I'm sure for our competitors as well.
Okay, so definitely the answer to that question is that yes, in industry there's a lot of opportunities, what are in general your views about the prospects of developing a career in food science? Is there upward development easy or difficult in comparison to other careers? Let's maybe ask Joanna.
I don't think so. There is like a single port. I think there's multiple ways to enter this field. We can see first regarding our panel that we have different type of career, and we are all or have been involved in food proteomics. So yeah, I think there are multiple ways to enter this field. It can be like with academic industry. We said that there's now more and more implication of proteomics in the food field. That's something I noticed while working on this project. That for the beef meat industry, for instance, there were a lot of biomarkers that were discovered, but none of them were really further investigating in order to develop a proteomics-based tool. So I think this is not just for the beef meat field, but it is more general to all field related to food. And so there's, I think, a lot of opportunities in this regard for food proteomics.
Thank you. I actually have another question more relevant to food science in academia. Blandine Chazaroun is asking, are there any unique challenges specific for food proteomics, food science research in academia? Maybe Melanie could answer that question.
Yeah, sure, I can jump in on that. I mean, I think there's always challenges, right, in getting funding for academic research, et cetera, right? And I think a lot of those maybe kind of relate to good strategies are similar to what Joanna described, right? Is finding a collaborator, right, to kind of have the end application, right? It's hard as a proteomics person to be a expert on all the applications of proteomics, right, for all different types of systems. So finding good collaborators, right, that can, that are able to have a relevant question, right, that then can be answered with proteomics is really kind of key, I think, to maintaining a good academic program and getting consistent funding. So yeah, and I mean, just to the previous question, maybe a quick comment as well. I think, just from the outside, from outside the industry, I think, food proteomics is still a little bit, well, quite a bit underutilized, but really on the upslope, right? Even just in the past couple of years, I think we've seen more concerted interest from industry companies when they can see how it can be useful and implemented in a pretty streamlined way, which I think both Jamie and Joanna kind of commented on, that it's underutilized and they need expertise, and is kind of one of the key holes. So I think there are career opportunities, especially now. Maybe 10 years ago, I probably wouldn't have said that, but
right now
we're on the upward part of the curve.
I have a very similar feeling. Yeah, it's partially why we also decided on that topic for this career series. I see Corey has raised his hand. Would you like to add something onto the previous questions?
Yeah, so I guess I wanted to reflect just a moment on Nathan's question about career prospects. And there's something kind of immediately topical, right? Which is right now the biotech industry is hot like I've never seen it in my entire career. So I think anybody who's spent time in front of a mass spectrometer becomes hireable in a way. So I mean, I think this is a great time if people want to look at ancillary areas kind of outside of kind of core, right? I think so much of us tend to kind of say, okay, I do a PhD, it's biomedically oriented. Where do I look for a job? I look for a job at a Quest Diagnostics, or I look for a job at a Merck or a pharma company. And now, because the field is so hot and there's all sorts of things going on, I think there's a lot of interesting opportunities right now in a way that there hasn't been in the past. I think it's probably still true that the very best candidates in the space will have gone through a traditional biomedical program, right? There's going to be fewer proteomics programs centered in food science departments and things like that would, kind of feed all the demand, right? So, again, I think there's fantastic, opportunities. I think the one thing I would say, related to it being a challenge in food science research and academia, and this is a picture, again, that I bring from when I was all the way back, particularly in Melanie's, discussion about the fact that she works, at a land-grant university. A lot of food science departments are affiliated through land-grant colleges, and each one of those colleges has tended to kind of pick up the best agricultural, area, best agriculture that's kind of in their area. So I'll just give an example. When I was at the University of Wisconsin in Madison, it's known as a dairy state, right? And so a lot of money flowed into, just kind of, we had a pilot plant, for dairy food production. All the extension programs are around cheese, butter, ice cream, yogurt, production, things like this. So the research dollars also flowed into that direction. So if you have an interest, in dairy chemistry, if that's an area that you see a future, you'll find that the academic programs will be quite, at least it was that time. I think there's still some similarities, even today, in topic areas where the universities have got real depth, depth and strength. And so again, if you go to a place like Madison, Wisconsin, and you do proteomics in a food science area, it's probably going to be related to dairy. Not that that's a bad thing, but it does give you kind of a space that you'll work in that may be somewhat more constrained than if you come from a top-notch biomedical institution where you've just kind of focused 100% on proteomics.
That's actually a great tip, Corey. Just do your due diligence research in terms of the geographic geographic situation, whether your university or industry company that you're interested in, well, where is it located and how this area is funded. That's amazing. That's amazing. It's something that I would never ever thought about, but it's a great point. Okay, I do think there is like a follow-up question to you, Corey. Plant-based from Blandine Chazarin. Corey, plant-based proteomic approach is challenging. How did you overcome those for plant-based food?
Well, I have to say, I didn't spend a lot of time in that space, right? So my tenure at Impossible Foods was pretty short, but I would say a couple of things about the plant-based food area. It is blue sky, right now in terms of people approaching things like ingredient discovery and food functionality. Structure function relationships are still a mystery, frankly, for people that, have all of the tools at their disposal. And we're just kind of talking about, people looking at enzyme systems biology approaches, right? You ask a question like, why is cheese stretchy, right? Why does my plant-based burger smell or taste or have a textural characteristic that's different than the real thing? I mean, this is fundamental, kind of fundamental science, and so I think I would like to say that I don't have any great answers. The thing I can say is that mass spectrometry, frankly, is uniquely positioned in this space because the level of protein structural information that we can generate in relatively short periods of time in extraordinarily complex systems. That's an underpinning technology that you just can't really touch with many other approaches. So, thumbs up for mass spec.
Thumbs up for proteomics participants and mass spectrometrists here. So actually we have related questions in the chat box, in the Q&A box. What skills are you looking for? And would food science graduates have advantages over those who are in stricter chemistry biotech field? So maybe Jamie.
I was kind of hoping that one would be thrown my way. Yeah, so I would say, it depends. I would say that for a company like Cargill, there are two paths you can go. So I'm actually based in core research, which is a subdivision of Cargill in general, and our core research group in Plymouth, Minnesota. But we also have application scientists. We have, let's see, how do I describe this? Tech services scientists. So the tech service folks are the folks that deal directly with our major customers, namely McDonald's and anyone else that you can think of that buys our eggs or our citric acid, right? So that was, I would say, my sort of path has been in core research, kind of more removed from dealing with our customers. So that I would say biology and chemistry tend to be preferred in that space. But if you have an interest in applications, if you have an interest in dealing directly with like a major corp's major customers, they do definitely prefer food backgrounds. So just an FYI, I would say though scrappiness. I wouldn't say that if you're in your career and you want to make a switch, I don't know what the demographics or the career history of the participants are right now, but I would say just as a personal anecdote, and this is what I was going to say earlier, is never give up, be scrappy, take any opportunity that you have, and you can dodge, duck, dip, dive, and dodge and make career changes. I mean, I knew nothing about proteomics 3 years ago, and I have a biology and chemistry background, but it showed a huge interest and acumen for the topic. So there's always hope.
All right. Do you, are there other participants, panelists would like to add on to top 2 or top 3 hot skills that would give you an advantage over your competitors in food science field? If not, then there is one more question about mass spec, and basically the question is, how much is it used in food quality control? Is it predominant technique, or are there other techniques that are specifically used in quality control? Let's ask Joanna.
I'm not really sure. Maybe other panelists are more able to answer this
no
question.
Problem.
Sorry.
Melanie, would you like to, or Corey? Melanie and Corey?
Yeah, I mean, I don't know that I'm the perfect person to answer this, but I would say it's used very frequently for small molecule quality control, right? So things like aflatoxin and pesticide residues. And I mean, a lot of those are primarily MS these days because of the multiplexing capabilities, right? I would say probably less so on the proteomics specific front in terms of routine quality control. I think probably more so used, I mean, and Jamie could probably answer this question better than I can, but, you know, for process optimization, for troubleshooting, for that sort of stuff, rather than routine quality control. But there are some, you know, mostly small molecule applications where MS is the predominant workhorse for quality and safety monitoring. So I'll hand it over to somebody with more hands-on experience.
Before we hand over that question to Corey, I would like to ask a small follow-up question to you, because there, obviously you mentioned in your introduction proteins would be the main allergens, but there are other allergens as metals, nickel poison, nickel allergy. How would you tackle metal detection in food, or you have never had experience with that?
Yeah, I'll say our group does not work on that. That's not a primary route of exposure, right? So most of those are kind of cutaneous skin exposure type of scenarios.
Okay.
And they're also, you know, not regulated as a sensitizing agent, right? I mean, they're regulated for different reasons. Right. So the one other, we used to say all food allergens are proteins, right? We sort of had to amend that a little bit because of recent identification of alpha-gal allergens. I don't know if you've ever heard of these, but it's related to red meat allergies. I'm sure Jamie's familiar from a large meat processor. But anyway, you know, that's a carbohydrate group in particular that causes these reactions, but they're really in their own category that's separate from everything else that we do, and they're not regulated as food allergens either. So that's usually the caveat there.
Thank you. Corey, would you like to add on that quality control question?
Sure, just real briefly. You know, I think I agree 100% with Melanie, right? There are labs all across the United States that are using mass spectrometry every day to look at the quality of ingredients and food products, right? Pesticide screening is, you know, clearly a big one, using LCMS and GCMS technology. You know, those are going to be both internal to, you know, internal to companies, but also kind of government regulatory agencies. So again, lots of opportunities for labs that are just doing a lot of, you know, serious screening. And, you know, again, you know, a growth area in some way. It doesn't have much to do with food, although some of these things have become edibles, right? The growth of the cannabis industry means that there's been a lot of labs that have popped up to, you know, to look at these things. Because ultimately, you know, some of them are getting incorporated into gummies and beverages and all of these kind of things. Same thing, pesticide screening, basic compositional, you know, kind of analysis and characterization. So lots of aspects, not proteomics per se, though, really. One thing I was going to mention in the, you know, proteomics space is, right, I think there's still the ability for organizations to use the technology to move very quickly, right? So maybe there's an interesting question about, you know, what is the composition of some material that was used as an ingredient, right? It's meant to be beef. Does it contain pork? Does it contain chicken? You know, those types of things typically have been done by immunoassay-based, you know, technologies. But our food systems are becoming more complex. The questions we're being asked, what fraction of food might be used as an adulterant or an additive or an extender, you know, in some of these cases means that mass spectrometry has a role to play in terms of very rapid response, to answering some of these types of questions. But I think you're going to find that in specialty labs that are kind of right on the front lines trying to ask complicated questions, because again, if it is a question of how much chicken may be blended into a beef, kind of a labeled beef product, that's probably going to have been worked out by immunoassay. Just kind of broadly, what I wanted to do, and this is not, again, proteomics related, but in the chat, I've just put a couple of papers and a couple of links to some really interesting work that just goes to show the power in the use of mass spectrometry in analyzing these, kind of really phenomenally complex systems, when we're talking about food products. So anyway, I just wanted to let people know, if you're interested in something that I think is pretty sexy use of mass spectrometry in this space, more metabolomics picture than proteomics, to maybe take a peek at what I dropped in the chat.
Yes, I did notice that. Thank you so much for this. That's very helpful. And also, Jamie, thanks for mentioning a very important skill, which is the data analysis. The stats, the bioinformatics is extremely valuable in hiring, in proteomics fields in general, all over, not only food industry. I think we have time for one more question. There's an interesting question from Nate. What kind of food proteomics startups should we all be starting or throwing money at right now? I guess Nathan is looking for new avenues for his investments.
We need a disclaimer that no investment advice is given at the HUPO seminars.
That took some guts. Yeah. Anyone that would like to answer this question first?
I'm happy to take a stab at it real quick.
Sure, go ahead.
I guess I think there are two areas right now that, where the effort's really being spent, right? It is about protein replacement by fermentation, right? So people would love to be able to just crank up a fermenter and make a ton of protein, that's got the same kind of functional characteristics as something that's coming from a natural source, right? So people are really, really interested in that. We've heard about kind of, cultured meat, right? That might be production of cells, but really just protein, right? Can you make casein? Can you make casein or whey proteins that again have the functional characteristics of the material that we normally would get from taking cow milk and fractionating it? And I think the other thing is ingredient discovery, right? Which is to say, there's protein everywhere in this world. Most of it we don't eat, but that's only because it's in forms that we don't typically think of, being very palatable or interesting. But if you isolate proteins, you purify proteins from all sorts of sources, are they valuable, relevant, or interesting as food sources with unique functional properties, unique ways of harvesting, low cost of production, all of these kinds of things. So to me, that's kind of the two areas right now that kind of predominate kind of the food tech in industry space right now.
How about food supplements?
I have to say, I don't know so much about that space.
Okay, I see Jamie raised her hand. Would you like to add something on that?
I agree with everything Corey, said. I think that's dead on. But I would add, in general, just, as I alluded to sort of in my little intro, I think animal health, human health, how do you make shrimp get fatter, stay pinker longer? How do you make a cow less gassy? How can you feed additives, different feed substrates, across the sort of mammalian platform, I think is very interesting. What do bioactives, peptide pieces, and what defines a bioactive? Clearly you've got small molecule interplay with those sort of shorter chain peptides, but sort of what do they look like? Can you find a novel one? What does the IP space look like around there? I think that's a really cool space that I think a lot of companies are fighting each other to try to figure out how to own and where to go with it. But I think health in general, including human health, That gets, complicated at a company like Cargill, where we're focused a lot on animal health. But I think we're having some movement there, and that we might start to maybe look in that space, because then there's a whole lot of different regulations that actually come into play if you start to do any work aimed at humans. So those are the spaces. It's healthy. The healthier we all are, I think the better we're all doing.
That's actually something super interesting, given the fact that majority of the drug sector is sales are going into animals. I remember seeing some documentary about it a few years ago. I think it's like more or less 20% goes to humans, 80% of the sales goes to animals in pharma industry. So yeah, that's actually amazing. All right, we have 4 minutes left. I see there is one more question, so maybe we can quickly answer that, and then I have few closing words, few closing remarks that I would like to make before we finish. So Ximin Chen is asking us, do you have any suggestions for someone who is currently looking for opportunities in food proteomics? I guess that would depend on what stage maybe of the career are you at, but let's assume this is early stage, like maybe graduate, PhD, postdoc stage.
I would say go to LinkedIn, search for the names of the companies that are out there kind of in the food tech space, right? Because I think you'll quickly build a web. If you look for Beyond Meat, if you look for Impossible Foods, if you look for Just Foods, if you look for Climax Foods, right, there's kind of a number of kind of companies that have been around a few years, some companies that are brand new. You'll quickly find a web of companies that are kind of involved in this space. And I think, starting there will allow you to touch, kind of the broader community. And then I think, Jamie's at Cargill, right? You can think of the names of the stores, the names of the companies you walk up and down your, kind of a supermarket aisle, right? General Mills, M&M Mars, those are all companies that are going to have a fundamental interest in science and food science and use the best technologies, right? So I think those are all good places to go take a look at.
Thank you. That's a very useful tip. So finally, I would just like to give a few announcements. So we are the members of ECR US HUPO committee, myself and Cora and some other participants in today's event, like Nate, like Lindsay, and Maya and Rocky. We are currently working on some other very interesting activities for any ECR interested. And I would like to announce that on February 26th to March 2nd, US HUPO conference is taking place in Charleston, South Carolina. And if you are interested in our mentoring days, in our ECR mentoring day activities, or in our social activities, such as actually pitching some job offers, please join us. There is the registration rates are going to increase very soon on January 26th. So please save some dollars and register before that date. And basically what we have in store for you on the mentoring day that we are working on, it's a very, very interesting discussion panel about time management, project management. And we would like to share all the experience and resources that are out there for you that you could use in your everyday work in the lab to organize your work efficiently and your time efficiently. there is a plethora of tools to do that and strategies to do that. And aside from that, we are going to make another workshop about social media presence, professional presence at the conference, tricks and tips for young scientists to help them basically pop up in the field or just gain more visibility. And aside from that, we always organize at the conference some social activities. This year it's going to be in person, so we'll see how that goes. It's going to be one of them is going to be lab company pitches, so it's just kind of job opportunities event where you will be able to mingle with people who are interested in hiring you. And also we will do kind of an elevator pitch event where you will be able to learn or see how to sell your project, your ideas to other people. So we again warmly invite you to register for the US HUPO conference and hopefully see you there. We are coming. We are very, very excited. It's an in-person conference. For me, it's going to be an in-person conference, first one in 2 years. So I'm really excited. Okay, thank you to our panelists today. This was amazing. Thank you so much for joining us today. And thank you to all the participants, and have a nice rest of your Wednesday. Bye.